Okay, well, welcome back. Alexandra and I are, I guess, first grateful to all the people that lectured, so also including you. So let's thank them again. And then we wanted to, sort of as a warm-up for the conference, which begins this afternoon, have the opportunity to hear from some of the students and postdocs that are attending. And so we have selected 5 titles from the posters that were submitted. And these will be 10-minute talks. And I won't speak much more. I'll just introduce Fabiola Garcia Fernandez, who will tell you about her work.
Okay, so good morning. It's a pleasure to be here and have the opportunity to share my work with you today. So in our lab, we are interested in the study of multi-scale chromatin dynamics upon DNA damage. So to briefly remind you, one of the mechanisms of the DNA damage response is the remodeling of chromatin around DNA lesions, which allows the access of all the repair machinery to the broken DNA. And it is known that chromatin undergoes dynamic changes in its structure before being completely repaired. Sorry. Okay. So here, okay. Before being repaired, which are mainly triggered by post-translational modification of histones. For example, the first remodeling event that occurs upon DNA damage is a really rapid and transient chromatin relaxation, which is followed by a longer phase of compaction. And some studies have suggested also a second phase of relaxation, which seems to be needed to complete repair. All of these remodeling events are shown so far at the nuclear scale, at a large scale. However, we don't know exactly what happens to chromatin after damage at lower scales of its organization. We know from studies mainly done in yeast that the chromatin fiber is more dynamic after damage, which seems to favor repair. However, in human cells, there is no consensus to describe how chromatin motion is altered after damage, taking into consideration time and also space. That's why the main objective of this study is to characterize the early remodeling events that occur at the DNA damage immediately after DNA damage, at multiple scales of chromatin organization, and to understand their mechanisms and also function. So for this, we established an original setup that combines microirradiation and multi-scale chromatin imaging in human cells. So we installed in our resolution microscope, super resolution microscope, a UV laser that allows us to irradiate single cells, triggering single and double stem breaks. So then, by using some imaging techniques, we could monitor changes in chromatin conformation and also dynamics immediately after irradiation, and at several scales. So, for example, from the nucleus to the fiber and at the nucleosome scale. So first, we start by corroborating this chromatin relaxation in our setup. So for this, we use cells that express histone H2B tagged with a photoactivable GFP. So by irradiating a specific region inside the nucleus of these cells, we could simultaneously induce DNA damage and photoactivate histones only inside the irradiated region. And we observed that there is an increase, a rapid increase of the size of this irradiated area, of the photoactivated area, which indicates chromatin relaxation. So we observe a peak between 1 and 2 minutes after damage, and then chromatin starts to slowly recondense, reaching its initial compaction levels 10 minutes after. So to know if this relaxation affects chromatin dynamics, we first look at the dynamics of the chromatin fiber. So for this, we use cells that harbor a LACO array in chromosome 1. And we track the dynamics of the fluorescent locus after irradiation of a neighboring region or of a distal region. So we plot here and we calculate the mean square displacement in function of a time interval. Before irradiation in blue, after nearby irradiation in red, and after distant irradiation in green. And next to it, I put the representation of the 500 first points of these trajectories with the same color code. So the MSD and also the trajectory show that compared to the undamaged condition, the locus explores a larger nuclear volume only after a nearby irradiation. So to go further, we fit these MSD curves with some diffusion models. And as expected, before irradiation, so the blue curve fitted really well with an anomalous diffusion mode. However, 1 minute after nearby irradiation, the curve cannot be fitted with a single diffusion mode, but at short scales, it follows anomalous diffusion, and at longer time scales, it shows directive motion. And to know if this effect on chromatin dynamics goes beyond the fibers, so for example, at the nucleosome scale, we follow the dynamics of histone H2B. So this is a kind of data that we worked with yesterday during Simon's lecture. This is an example of an acquisition of a nucleus expressing histone H2B, tagged with ALO and bound to photoactivable GFs. So to know what happens only inside the irradiated region, so after damage, we focus on histones inside this region, and we follow their dynamics before and at different time points after irradiation. Okay, so here I show you the displacement histograms, which represent the distribution of all the H2B traces according to their displacement in micrometer. Before irradiation in blue, and after irradiation in red for the different time points. So as early as 15 seconds after irradiation, we can see that the red histograms shift towards the right, which means that H2B is more mobile. And this effect is more pronounced 1 minute after. But we can see also that 5 minutes after, histones start to recover their initial dynamics, which is completely recovered 10 minutes after. So this indicates that H2B is transiently more mobile inside the irradiated area, and we corroborate that this is a specific response to DNA damage, since there is no change in histone mobility in regions outside irradiation. So to better visualize this effect on histone dynamics, here I put the representation of all the traces inside a nucleus before and 1 minute after irradiation. And all the traces are colored according to their diffusion coefficient. So red traces show a slow motion diffusion, and the blue traces show a higher diffusion coefficient. So we can clearly see here in these zoom images that 1 minute after irradiation, there are more blue or fast histones traces compared to the undamaged condition, which we can see that there are more red traces. Okay, so we saw together that this rapid and really transient chromatin relaxation is accompanied by the also transient increase in dynamics, which occurs from the fiber to histones. So then we wanted to know what is the mechanism of these multi-scale chromatin remodeling events. So I invite you to come to my poster, I think on Thursday, to see this mechanism. So thank you very much, and thank to our collaborators.
Questions?
Hi, Fabiola. Do you interpret the increase in mobility of H2B as a release of the histone from chromatin?
Okay, this is a question that I always have. For us, it's not a release of histones, okay? It's only the increase of mobility. And I didn't say, but we work with Sébastien Huet for this part. And in his paper in 2017, he showed that, I think I have it here, sorry. Yep. So he labeled DNA, and he saw that DNA moves away the irradiated line with a similar speed to that of H2B expansion. So it means that there is no histone release. And from our SPT data, we have the similar number of traces inside the irradiated region before and after. So we don't believe that is a really release.
Very related question. Like, do you think, when you have a polymer and you have an end, and you look at something which is just at the end of the polymer instead of in the center, it will move more. And so since you do DNA damage, it might be that there is also the effect of losing the constraint, having some free polymer ends that can diffuse, and they will diffuse faster. Do you think this could account for this?
So do you mean that the free histones?
No. Like the polymer that is cut.
Yeah.
Then automatically, just because of that, maybe, will move faster. Because, when a monomer, like an histone, is linked to both sides, it will move slower than an histone that is linked only to one side.
Of course. Yeah, this can be possible. So I don't know if I can answer your question with this, but we, for example, for the LACO array, we measure the distance between the LACO and the irradiation, and after damage, after irradiation, there is an increase of this distance. So it means that the LACO is like moving away, and then it recomes. Yeah, I think there is this repulsion.
I think we're recording.
Very interesting. So do you have an idea of how much DNA damage you're generating? Like how many open ends you have?
No, this we don't know exactly. Okay, we know that we trigger single and double stem breaks. This we can measure, for example, with the recruitment of different proteins, PARP1 or 53BP1, but we don't know exactly the number of breaks. This is something that it's hard to say with the irradiation.
Thank you.
So you mentioned directed motion, right? So have you looked at if the directed motion is like oriented outwards of this irradiated region, or is it directed in certain directions?
Yeah. So as I said before, so I measure the distance between the irradiation and the LACO, and it goes really directive away, like here, like directed motion, and then it comes back. But yeah, we need to exploit more this MSD.
All right. Thank you again.
Okay. Thank you.
And the next speaker is Ronaldo Oliveira.
Hi everyone. I want to start first thanking the organizers for inviting me for this talk, where I have the opportunity to show our work. And first, what we want to do generally is use a polymer model, a minimal polymer model, to understand chromatin architecture. But first, let's talk about a little bit of chromatin organization. The chromosomes inside the nucleus are organized as territories, as you can see here, But within these territories, it can be organized as compartments, as it was beautifully explained by Dr. Sumitaba early in this workshop. And these compartments, we can say that they could be A's and B's, as it was shown. But within these compartments, we also have another level of organization where these chromosomes, this polymer crumble to itself. It condenses mainly by protein complex, which is mainly by many protein complex, as you can see here. But so we have here the compartmentalization, which is shown here on the left, which would be one of the mechanisms that could drive this chromosome organization. And also we could have this protein-driven motorization given by many protein complex like SMCs and CTCF. And all of these mechanisms can be seen in the high-C experiments, as we've been seeing these last 2 days. So if you think about chromatin or chromosome organizations mainly by these two mechanisms, I want to define a polymer model and to understand what are their individual contributions to this organization. So this would be the main point of this talk. And for that, we're going to use the minimal chromatin model that was also explained during this workshop. And for that, as we have the high C, we're going to train the parameters of this model on top of this high C using the maximum entropy approach, which was, we did that before, we did that during the early workshop. But just as a reminder, our first assumption is that our polymer has, our Hamiltonian has a term. This first term is the polymer, which takes account the polymer nature of it. And the second would be the term, the type-to-type term, which takes into account the compartmentalization, these interactions between the different types of the beads. So it's the compartmentalization. And the third term, which was also explained during the talk, is the ideal chromosome. So this is like a lengthwise compaction term, which is the protein-driven motorization. So in this model, let's just stop here and just think about these two main terms, which is the compartmentalization, the type-to-type, and the ideal chromosome. We want to investigate what are their contribution to the model when we have the configurational ensemble of this simulation. So for that, we're going to do two approaches. First, we're going to learn the compartmentalization term, which is types, and then the ideal chromosome, which has been done before. And now we're going to do a second approach, which is the opposite, which is trying to learn the ideal chromosome first and then types. As you can see here, if we train types first, we have a baseline. Where's the mouse? If you can see here, when we train compartmentalization or the type-to-type term first, we have a baseline here which is close to -0.3, which would be the average attraction of the beads of the different types of the chromatin loci. And then when we go to the lengthwise compaction, it suddenly goes to 0. But the first term, the first beads interactions are quite strong. But if we do the opposite, then this baseline moves from the types and goes to ideal chromosome. So the compaction, the lengthwise compaction term now is stronger. And then suddenly, if if you train types later, then you have like a fine tune, and then the energies, the average energies are slower between the different beats. And this is here, and here is just to show you how the trained potentiometers compares with the experimental high C. So in the bottom triangle, you have the computational ones. So now we have two trained models where in one the attraction, the compartmentalization is stronger, and in the other one you have the lengthwise compaction is the one that's dominating. And here, as we have a simulation of a polymer with these different types of interactions, we can generate ensembles of structures, and then we can analyze these ensembles and create assumptions. So here is just to show to you that after this training, I have two ensembles. This is the first approach, and the second, and here it's a projection along the principal component analysis, and I'm comparing with the homopolymer model is where we don't have those specific type of interactions of the chromosome. Similar to what Sumitabo explained, but he had three types there, A's, B's, and C, and here we don't have, it's just the homopolymer part. So this is just to show to you that this different approach will give you similar ensembles. Of course, that along this axis here, it has a separation, but roughly speaking, the ensembles are similar, as I'm gonna show you in this next slide, where I compute the similarity between the ensembles with a non-measure in the field, which is Q. Q measure the similarity between the different structures. And then when we compare the relativity entropy between the ensembles, it's mostly the same, like the two approaches for training the chromosome interactions. But what I want with this, as I mentioned in the beginning, the chromosomes are organized as territories, and we had this caveat in the model. When we use the previous trained model, the interactions between the different chromosomes or the interchain interactions were underestimated. So with this new training, I'm gonna show it to you that when we run the multiple chromosomes, when we want to represent these territories, we have the same scaling, the same genomic scaling, as Alex explained to us. The average contact probability along the sequence is the same for the interchain. But as I mentioned, the interactions between the different chromosomes are different. So what we see here is that with this type of optimization, the chromosomes are more spherical, but with the lengthwise compaction more stronger, we have a deformation of the different chains, as you guys can see here. And another thing that I want you to notice is that the interface between the chromosomes, now instead of having all of the B's interacting like this, we also have some A's in betweens. And what I want to get with that is the representation of the interchain interactions. So here, with the original micron, we have an overestimation of the interactions between A's, which are the loci that are more exposed. But then when we compare with the experiment like this, so this is also overestimated. But with the new training, as we can see here, the A's are the interactions between A's and interactions between B's are closer to the experimental values. So this is first attempt to represent the interactions between the different chromosomes. It's not perfect. We can make a lot of improvement here, but at least we have a better understanding of the different types of the different mechanisms that the model can handle, like the compartmentalization and the motorization. And with that, I want to conclude that I also did lots of different parameters optimization with this model, and roughly I got the same average energies, so different routes leads to ROM. This is a very good point of the model, and also with this stronger ideal chromosome, we can have a better improvement in the representation of the interchromosome contacts, which gives us like a balance between phase separation and the territory formation. And I would like to thank Professor José Nushik for the opportunity to learn how to work with the chromatin, and also his team, which is great. Vinicius Contesoto, Antonio, Oliveira, they're both here. They were the monitors in the previous workshop. Sumitavo Brahmachari, Mateus Ragini, and also my lab in Brazil and all of the collaborators. Thank you very much.
Time for a couple questions. Well, maybe I'll start with one, which is that it seems like with the two approaches, one, you're doing training of, I guess, as I understand it, lengthwise compaction first, and then the compartments, and then vice versa. And so you see some differences between the two approaches. Is there like I don't know, a clear way to understand why you see these differences in the resulting model?
Yeah. For the single chromosome, I didn't see that much of the difference because the average interactions between the beads, it's around -0.3, which is mostly the average of the interactions. For the single chromosomes, it doesn't matter if you do one or the other, because in the end you have this average interactions between the beads. But suddenly we saw the difference when we use multiple chains. So we had this underestimation of the interactions between the chromosomes, right? For this model, so single chains, it gets good results with the experiment, but for the multiple chromosomes, we had this caveat. So with a stronger ideal chromosome, the territories get a little bit distorted, and then you have, instead of having just B's in the interface, you also allowed, this distortion also allowed some A's to get inside, and that starts to correct the interactions between the different chains. So, of course, this is just a first approximation of a nuclear cell, because it has just 4 or 5 chains, where we would need like 23 pairs, right, homologal pairs of chromosomes, which would be a whole chromosome simulations. That we also have a poster about that, but that would require lots of more investigation. So pretty much is this single chromosome, you don't see that much difference, but for multiple, you will start to see it.
Yeah. Have you tried or have you thought of comparing the output of your modeling to some imaging technique, maybe live cell imaging or maybe static imaging?
No, I didn't try with imaging yet.
Do you think about it? Do you think it's a good approach?
Yeah, we have an ongoing project where we are trying to use imaging data for chromosome simulations. So it's an ongoing project. It's really in the beginning, but it showed some promising. But in that model, instead of using the previous trained types and ideal chromosome, we expect to use the 3D information of the image and from 3D generate the 3D conformations instead of the regular model, which use 2D, which is high C trained parameters to get the ensemble averages to 3D.
Okay.
So that will be like a good way, a good direction for these projects.
Okay, thank you.
No problem.
Okay, we can probably take one more quick question while the next speaker sets up. That'd be Paulina. Well, otherwise, thank you.
Okay, so hello everybody, my name is Paulina. I'm a third-year PhD student in Fadenberg Research Lab, and I'm very excited to share my research with you on deciphering the gamma of genome folding using a deep learning model, Akita version 2. So, as we know, CDCF is a zinc finger, a DNA binding protein that's extremely crucial for forming TATs, and it binds to DNA in orientation-dependent manner. And in my presentation, I'm gonna depict it as an arrow, as in this bottom picture. And the core motif is around 19 base pairs long, so it's pretty long motif. And as you see on the right, there is a CDCF ChIP-seq and a line high-C map. So when you look at the boundaries and like trace above, you can see peaks of CDCF ChIP-seq, but it's usually more than one peak. So there are clusters of CDCF sites. So kind of a challenge is to identify which of those CDCF sites is really important to create this particular boundary, or maybe all of them are, or maybe a subset. We really don't know. So there are depletion experiments in which CDCF have been acutely depleted. And we can see that globally, CDCF is extremely important to create TATs, and they disappear if CDCF is not in the cell. However, it's a global picture, and we cannot really see the impact of particular CDCF sites on the formation of boundaries. So we know from literature that there are some kind of deletions, insertions, disruptions of TAT boundaries or particular CDCF sites, and some of them doesn't cause any kind of weird phenotypes, but some of them can, like for example this EpiHA4 locus. It's connected with limb phenotypes like brachydactyly and F syndrome. However, still in this picture, we don't see the effect of particular CDCF sites if, for example, the entire TAT boundary is disrupted or disappear. So this way, we come to Akita. So ideally, if we have a magic way to get the map from DNA, we can disrupt DNA and see the impacts on the map. And this magic way, it's gonna be our neural network. So we live in neural network era. There are multiple neural network models that like pave the way for Akita, and there are multiple models that take DNA sequence and produce some kind of function or track, like ChIP-seq or RNA-seq. So mathematically, you would like to have a function that takes DNA sequence as input and outputs contact frequency map. And this function is going to be our model. So precisely, Akita is a convolutional neural networks that kind of catch patterns in DNA sequence and associate them in patterns and contact frequency maps. And actually, the output is a function of contact frequency map is logarithm of observed by expected. Okay, so our goal is to extract general principles of mammalian genome folding. And how we're gonna do it, we're gonna make millions of predictions and try to figure out some kind of gamma with focus on CDCF sites. So in silico experiments give us a lot of flexibility in how experiments can be designed. So in my experiments, we started from generating neural backgrounds that are obtained by shuffling real genomic sequences by 8mers. And so here on the left, you can see a genomic sequence with CDCF sites with their flanking sequences depicted above. And after shuffling, we kind of get rid of those CDCF sites, and we also get rid of patterns in the contact frequency map. And then in the sequence, we can reinsert CDCF sites in a more controllable way, and we can see like really clear boundaries exactly where the CDCF sites are inserted. So, and in this kind of setup, we can change a lot of parameters and measure how they change the predicted map. So the first experiment, we focused on flanking sequences. We were curious how the flanking sequence length is going to change the impact, or the maps are going to be stronger or weaker, what we could expect. So here I'm depicting visually the first experiment in which the same CDCF motif with their genomics flanking sequences have been inserted, and the flanking sequences were getting bigger and bigger. So this is just one example of maps for one CDCF site. So visually and intuitively, we can feel that the flanking, the bigger flanks give us the more intense color on the maps. To quantify it, we created a score we called insertion score. So if we have a map to quantify, we subtract the background map, we take a square of this, we sum it up, we take a root, and the value is insertion score. And intuitively, the higher values, the more colors on the map. So here I'm going to plot the impact with respect to the flank length. And so I tested more than 7,000 of CDCF sites from the mouse genome. And to depict the results in more like intuitive way, I split those sites into 5 groups with extreme, strong, considerable, mild, and weak impact on the maps. But first, I'm going to show you single tracks for like one line is one CDCF sites, and they're all from the top extreme group. So you can see that even in this very clean experimental setup, we still see a lot of heterogeneity between CDCF sites. And this is the average for the entire group. And now for readability, I'm going to remove the raw data and plot all the other 4 groups. So as we see the flank length, with the flank length getting bigger, the impact is getting higher. However, this impact saturates. And we see this effect for the top groups, not really for the weak ones. And please note that actually huge percentage of the sites that I tested are actually very weak, and the impact is actually, like predicted by Akita, is negligible. And the impact saturates around 15-20 BP of flanks. Those flanks are length is symmetrical, so it's 15, 19 core, and 15 again on the other side. So then seeing how different the impacts can be for strong and weak sites, we've been curious if weak flanks, for example, can work with strong cores, or like if there is any rule of incompatibility between them. So if we depict this problem with only 3 CDCF sites and show it as a matrix, we actually see that we have the results for the diagonal of this matrix. However, we're lacking the off-diagonal values, so we don't know how strong cores work with weak flanks. Do they even can work together? So I performed this experiment with 100 strong, 100 medium, 100 weak sites. We separated cores from flanks and we shuffled them around. So we tried thousands of combinations between them. And this is the results, a huge 300 by 300 matrix. And the first conclusion we can get is that there is no strong diagonal. So actually cores and flanks can work equally well with like not their own pair. And also, if you just focus on the first, like 1/3 row, rows of the matrix, we see that for strong cores, the strength of the entire strong and plus core side can change by a lot depending on how strong the flank are. So actually we were kind of positively surprised by how huge impact flanking sequences might have. Okay, and we also compared our intuition of this additive effect between cores of flanks. So on the left, there is a matrix we got. So those are the values predicted by Akita, where we were testing cores and 30 BP flanks with the effect of sum of the strength of cores and strength of flanks separately predicted by Akita. So those matrices are pretty similar, if you agree with me, with the same color scale depicted. Okay, so there's more of our results. I just don't have time to show you all of them, but please come by to poster number 24 on Thursday. So here is a short summary of all the results that we have in very this simple depicted way. So as we saw, the longer flanks, the bigger impact we see on the maps, but there are no general rule of incompatibility that we saw. And the bigger number of CDCF sites, also the bigger impact, which agrees with the observation about CDCF clusters. Then we also changed the distances between CDCF sites inserted in pairs, and the bigger distance, the lower impact. However, there are pretty big differences between orientations, with the biggest values for divergent orientation. And we also tested if there is any kind of specialization in CDCF sites, and we really don't see any. So the strongest dots we get on our predictions, the stronger boundaries those CDCFs can produce. So that's all of my conclusions. And in summary, I would like to thank my lab, especially my mentor, Jeff Fallenberg, for all support and guidance. And of course, Alexandra and Ed for the opportunity to talk today. Thank you so much.
Thank you. Questions? There's Jose in the back.
Cool. Thanks a lot. Very cool talk. Have you looked into what actually these flanks contain? So do they contain then other sites for CTCF, for example, or any other transcription factors, or what might be the reason or biological relevance?
Okay, so actually the set of CDCF sites that we tested have been filtered first, so we didn't want our CDCF sites to overlap each other, so they cannot really overlap another CDCF sites. We try to find with very different softwares any transcription factor sites that they can be. We really don't see anything. We see some strong, some sequence preferences that sometimes the strong sites have more G's here and C's somewhere else, but there's no really another transcription factor binding site there. For example, we saw maybe some of them have this sequence in 20% of sites with comparison to background, so it's really weak result. And we also try to reorder top sites, trying to find some kind of clusters of letters, but there's really nothing strong enough to call it a motif.
Okay, that was a very, very nice talk, but I'm just curious about how much you are limited by your assumptions, and in a sense that basically you assume that you're just putting your DNA as an input, and then you're looking at the maps and try to map this with CTCF. When you get the end, we know that not all CTCFs are involved in loops. So the percentage of CTCFs are involved in loops, not in loops, do you get the right number when you compare to experimental numbers? And also, there's a large percentage of loops that are not CTCF mediated. They're mediated by something else. How do you separate that when you just look at the data?
Okay, so I think in this project we have been focused on CCF sites and their impact on loops and different features on the maps. So the limitation of this study is definitely that it's the non-dependent of CTCF structures are not covered here. And definitely another limitation is that like everything what I'm presenting and like the Akita could produce is based on what Akita can extract. So if there are like more of the grammar or more of different elements impacting the genome architecture, but Akita couldn't extract it from the data, we really cannot see the effect here. And
Rob, your grammar is just all CTCF based. You don't give any other options, right?
yes, yes.
Maybe just one more question while our next speaker, Gregely, sets up.
Yeah, great talk. So is this based on a specific cell type, or this is an average of all the cell types?
Okay, so I'm working with version 2, which is actually co-trained on both human and mouse. So
No, but I'm talking about tissues.
yes, so there are 6 cell types, mouse cell types that the model has been trained on, but in our research, we weren't focused on any of them really. We usually average over all the cell types. So it's like average mouse prediction, I'd say.
And this is actually very interesting, but what is the variability in the looping patterns in these different 6 types?
Oh, okay. So usually the predictions are pretty similar. Sometimes there are differences in values, but we cannot really say if it's a difference because some of them were micro-seed data, which may give you like higher values, but usually the patterns are very, very similar. But the model hasn't been really trained to like make the predictions like extremely specific to cell types. If it does. There are other models that can be more like specialized in particular cell types.
Yeah, so what I'm trying to ask is, the 6 types that you're using, are they in the data? Are they pretty uniform in their patterns of looping? Because, for example, you have immune cells. Those that have their own very specific looping patterns. So I think something that you might want to consider that.
I think that like majority of the data was like embryonic stem cells or fibroblasts. So I don't think there were any immune cells that the model has been trained on.
Thanks.
Well, let's thank the speaker again.
Okay, so thanks. Welcome everyone. My name is Greg. I'm from Luca Giorgetti's lab from the FMI in Basel, Switzerland. First of all, I would like to thank the organizers for making this meeting happen and inviting me for giving this short talk, and to talk about my PhD project, which revolves around establishing a parallel reporter assay to test the structural determinants of enhancer-promoter communication. So just a quick reminder, as we all know, enhancers are cis-regulatory elements that can activate promoters from up to 1 megabase away in the genome, and they regulate the activity of promoters in a cell type and tissue-specific way. However, it is also well known that a promoter can be activated by multiple enhancers, and an enhancer can activate multiple promoters, so giving rise to very complex regulatory mechanisms, so that allows us to develop into complex organisms. However, there must be certain rules that really define which enhancer activates which promoters, because it is also well known that there are enhancers that activate not the closest promoter, but skip one and activate the next. So the question is, what are the constituents of enhancer promoter communication specificity? And of course, there are already some putative factors described in the literature. First of all, I would like to talk about biochemical compatibility, which revolves around the sequence determinants of the specificity. So basically, this states that the transcription factor binding sites that are encoded enhancers and promoters need to be somehow compatible with each other, so that these transcription factors can bind and activate gene expression. And evidence for this originally came from Drosophila. However, more recent studies in mammalian systems have reached somewhat contradicting results, suggesting that there might be broad compatibility or broad specificity between enhancers and promoters in mammalian systems. So it's still not really a question that is very well understood. as another factor, in our lab and in Wouter de Laut's lab, we have shown that enhancer promoter genomic distance also can regulate gene expression. In our case, we have developed a system where we could place an enhancer at many different positions in the genome compared to a fixed promoter. And what we've seen by measuring the promoter transcriptional output is that the enhancer promoter genomic distance quantitatively defines the transcriptional output of a promoter. And this result kind of suggests that an enhancer that is close to a promoter might be more likely to activate that promoter than a further one. And as another point, as we all know, the mammalian chromosomes are organized into topologically associating domains on the submegabase scale, but also within these domains of preferential interactions, enhancers seem to require spatial proximity to their promoters for activation. And CTCF and cohesin and also other factors have been suggested to mediate these interactions, but what are their exact contribution to gene expression is still unknown. So to study enhancer promoter specificity, previously many labs have used parallel reporter assays that employed a library of enhancers and promoter sequences encoded next to each other on a plasmid library. And this way you can study thousands or hundreds of thousands of different sequences and combinations. However, since they are on a plasmid encoded very close to each other, we cannot understand the contribution of chromosome structure. So therefore, this is a great limitation in the field, because there are results that suggest that indeed chromosome structure might be a very important determinant of enhancer promoter communication specificity. So in order to address this question, we need a completely new assay where we can not only investigate a library of enhancers and promoters, but also have a library of CTCF binding sites to be able to control chromosome structure between them, and also to separate them by a meaningful distance of tens of thousands of base pairs. And this is exactly what I've set out in my PhD, to set up such a library system where we can integrate enhancers, promoters, and CTCF sites in the genome separated by a distance, and then measure the transcriptional output and the chromosome structural features of these large number of enhancer, promoter, CTCF variants using next generation sequencing. And so with this system, I would like to answer still open questions in the field, such as whether the modulation of enhancer promoter physical interactions via CTCF looping induces changes in gene expression, and if so, is it possible to rescue poor biochemical compatibility by forcing loops between enhancers and promoters? And I have to here note that the project is still very preliminary, so I'm going to show you more the method development part and just some very preliminary data that I've got. So yeah, bear with me. So to set up the assay, I wanted to avoid potential confounding effects of endogenous enhancers and promoters, and therefore I set up the assay system in a so-called neutral TAD, where there is no other enhancer, promoter, CTCF site, and this TAD, as you can see on this Hi-C map, has a generally simple architecture. And into this neutral TAD, I've integrated a landing site for recombinase-mediated cassette exchange, so RMCE, that allows me to integrate a library of different sequences with high efficiency. And this landing site contains a positive and negative selection mark, so we can enrich for cells that have undergone cassette exchange and contain the library of enhancer promoter variants. And after initial testing, I've shown that in one experiment using this BXB1 recombinase, I was able to integrate around 1,000 different large payloads into the mouse genome. So after integration, the enhancers and promoters, since they were originally contained in a plasmid, they are encoded very close to each other, so around 1 or 2 kb. So to investigate actually the role of chromosome structure, we need to somehow create a genomic distance between them, because otherwise they would be just too close and therefore be in contact all the time due to the polymer nature of DNA. And to do this, we thought to employ another recombinase-mediated reaction. And for this, we integrated a recombinase, an inversion site downstream of the original landing site, which contains these other second set of recombinase target sites depicted with these blue arrows, blue triangles. And also, this inversion site contains the same coding sequence of our reporter gene, and upstream downstream of it a very short distal index. And this proximal distal index, as you can see, we will be able to read them out using next generation sequencing, and therefore determine if a transcript is coming from a proximal enhancer state depicted here, or from a distal enhancer state after an inversion has taken place. So using this flip-base recombinase, we can actually distance the enhancer far away from the promoter. Currently the distance is 60 kb, so 60,000 base pairs. And not only it increases the distance between enhancer and promoter, but also replaces the proximal index and with this distal index. And after initial testing, I've shown that using an integrated inducible flip-base system, I can invert approximately 90% of the alleles in 5 days using tamoxifen. So I've kind of shown you some parts of the project or the whole essay so far. So I would just like to quickly run through the whole experimental strategy in a more linear format, so we're on the same page. So initially, the genome contains two sites, a landing site and an inversion site, 60 kb downstream of it. And then we create a library of enhancer promoter CTCF site variants. And each of these variants will be identifiable via a short DNA barcode that is placed downstream of the promoter, so it will be transcribed. And we identify these variants and their barcodes using nanopore sequencing of the plasmid library. After integration with recombinase-mediated cassette exchange, we can investigate proximal enhancer-promoter communication, and after the inversion with the flip-base recombinase, we can investigate distal enhancer-promoter communication. So in a single assay, we can investigate proximal distal communication of different enhancer-promoter CTCF site variants. And everything is done in a pool. So after all these genome engineering steps, I will have a pool of cells from which I can isolate RNA and DNA. And as I previously said, using next generation sequencing, measure the transcriptional output or the expression level of these different variants. And so I would like to quickly show you some preliminary results. So this is my proof of principle library that contains 48 different variants. So it either has strong CTCF sites before a promoter or no CTCF sites. It can have 4 different promoters, a CAG, which is a synthetic strong promoter, SOX2 and UTF1, which are endogenous promoter, or neutral, which is a neutral sequence which doesn't have any promoter activity, kind of negative control. And again, it can have strong and weak CTCF sites downstream, and the cognate enhancers of the 2 promoters and the no enhancer control. So just to give you an idea, we can have cognate pairs with or without CTCF site, non-cognate pairs with or without CTCF sites, just the promoter alone, etc., etc. And so here is basically the preliminary data, and I just would like to quickly run you through the important parts that we have noticed. So first of all, they are colored by the promoter, and what we've noticed is that the CAG, which is in green, the expression level of this promoter is much, much higher than any other 2, which is somewhat expected because this is our kind of positive control. Then if we zoom into the endogenous promoters without any CTCF sites, what we've noticed is that a strong enhancer, like the SOX2 enhancer, activates both promoters higher than, for example, the UTF1's cognate pair, the UTF1 enhancer. So this suggests that there might be a broad compatibility of enhancers, and they can activate other promoters as well. And now looking at the proximal distal data and comparing the enhancer and proximal distal way, we saw that the CAG, which is a very strong enhancer on its own, did not really respond to the enhancer being close or far away, whereas the endogenous enhancers, the endogenous promoters, SOX2 and UTF1, have seemingly reacted and reduced their expression when the enhancer was far away. And this is in agreement with our previous findings when we measured the distance dependence of the SOX2 enhancer promoter pair, where we also saw a clear decrease when the enhancer was further away. So with this, I would like to summarize. I'm developing a parallel reporter assay to measure the transcriptional effects of enhancer promoter identity and chromosome structure. And I've shown you some preliminary data that are coming up from this library. Of course, I will need to still collect more data to really show the differences. And importantly, I'm planning to measure also contact probabilities of these enhancer promoter pairs to correlate the transcriptional levels that we are measuring with sequencing with physical interactions. And for more details about how this setup works and also maybe more preliminary results, please visit my poster. And with that, I would like to thank again the organizers and my lab. So Luca has been a very supportive boss, always up for discussion. And also I would like to mention Sebastian Smallwood, who is the head of genomics facility at FMI, and he has also been a very great help for us. Thanks a lot.
Have time for a question or two?
Amazing work. So when you do the inversion, you increase the distance, but you also flip the orientation. So did you ever check whether that has an effect?
Yeah, it's a great question. Thank you for asking. So far I haven't directly checked. This is also in the pipeline, so we can just, so it creates the reverse complement, so it flips the sequence. So previous results from other labs and also our lab have shown that enhancers might really act orientation independently. So when we position the SOX2 in multiple places, we didn't see any effect regarding the orientation. So I don't think that this creates a large difference, or this would bias our results. But indeed, this will be an important control to show the enhancers in both orientation in the library to show that they really activate the same way. Thank you.
Do you plan to create another system with different distance between enhancer and promoters? This is the first question. And the second, there was a paper maybe a month ago or two that they tested different enhancer at very different distances, and it seemed that they found some kind of element that was supporting longer distances, like enhancer promoter interactions. Do you see that it is possible in your kind of setup to find this kind of distance-related elements around enhancers?
Okay, both great questions. So first of all, so now we set at this 60 kilobase distance, and in principle it would not be difficult to make another cell line with a different distance. But I think in the scope of my project and my PhD, this might not be feasible, but maybe someone will pick it up. So we decided on 60 kb because this seemed like a large enough distance so we can measure like contact probabilities meaningfully, but short enough that we will still see activation. Because, for example, if we go too far, already with the SOX2, like if we go to 200 kb, there is barely any activation from the SOX2 enhancer. And then we thought we should go somewhere lower because we want to measure this. So that's why we decided on it. In principle, it could be possible, but I think for now we will stick to this. And regarding the second question, yes, so there are very different enhancers out there. So some are really well defined and a single kind of locus, and if you delete it, there's no activation. Some are more spread out, and there are these enhancer helping elements. Yes. So I think in our assay, it would be difficult to actually measure these because these locations are very well defined how we set it up. So these are really elements that we can't really change. I think what would be possible is to place them into the genome like a priori or like first, and then do the whole setup for the experimental assay. But this would require extra steps of genome engineering, which might be difficult. So I think in this regard, our assay is not very flexible.
Okay, let's thank the speaker again.
So today I'm going to be talking about a story between my lab, which is the Mirny lab, and Ines Drenenberg's lab to study the unique and really cool folding of this organism right here, which is the silk moth Bombyx mori. So across eukaryotes, euchromatin and heterochromatin occupy distinct spaces within the nucleus, with euchromatin being represented here as A, heterochromatin as B. our current model for how these different loci segregate from each other is phase separation. When we look at compartmentalization, which is the structures that we call this organization on high C, we can observe it as this sort of checkerboarding of interactions. If we zoom in more closely, we can take a pair of two different regions marked A, look at their intersection, and find enrichment of contact. If we do the same for B, we see the same, enrichments between pairs of contacts, between pairs of loci. If we look at the intersection of an A region and a B region, though, we observe depletion. When we performed high C on the Bombyx mori silk moth genome, we indeed observed similar folding patterns, with A interacting with A, B interacting with B, and depletion of interactions between pairs of A and B. However, we also observed a third type of compartment, which we term secluded S. What you might be able to appreciate from all these different maps is that if you take two different regions, each annotated as S, and look at their intersection, they show no preferential contact. And they show very little to no checkerboarding overall. So if we zoom in and we identify the features that make S unique, we can start with 1. On the diagonal, S domains exhibit high degrees of local contact. 2, if we look at pairs of different S domains, they show no preferential contacts with each other. And 3rd, if we look at all of the compartment interactions for a given S domain, we see this smooth lane of depletion. So do S domains and S compartments form by the same mechanism that we believe to be forming A-B compartmentalization? To answer that, I looked at how two of these features, the S enrichment on the diagonal, shown here on X, on the X-axis, and the S enrichment off of the diagonal, on the Y-axis, how these two features change when we induce A-B compartmentalization to varying degrees. As you can see, as one value goes up, so does the other. When we compare this to the experimental value, we can see that S is very compact and very secluded, and really does not travel along this axis that we predict for the compartment profile of S. So if compartmentalization is not generating S compartments, what might be? Previous work from my lab has found that loop extrusion generates these local domains of contact, and through this process antagonizes compartmentalization. Loop extrusion and TADs can be identified on high C maps through domains of enriched contacts with flames or stripes at their edges, along with corner peaks at their edges as well. And if you look at the contact decay from high C maps in which cohesin is present or absent from mouse embryonic stem cells, you can observe when cohesin is present this characteristic shoulder in the contact probability decay that then goes away when cohesin does. So do we see features like this in the Bombyx mori map? We do, specifically within S. So here I'm looking at some TADs from humans, and here I'm looking at some S domains. We see similarly insulation points, flames, nested structures, and corner peaks. And if we look at the contact scalings for S, for Bombyx mori, which I've computed for each compartment type individually, you might be able to observe that in S, you also see a shoulder in this curve, which is evident over here on the derivative of the contact probability decay. And you can also see it in B chromatin, though it is more pronounced and leftward shifted for S. So with all this information about what might be driving the folding of the Bombyx mori genome, I developed an integrated polymer model combining both A and B monomer affinities and separations and loop extrusion, specifically tuning the overall abundance of loop extruders in A and B versus the abundance in S separately, and kept processivity, which is the average size of a loop generated by an unobstructed loop extruder. And here's what we learned. Specifically, each feature that I've defined for you already corresponds to a different parameter or relationship between parameters. Our first feature, which is this on-diagonal enrichment, can be best understood as the relative abundance of loop extruders in S compared to the rest of the genome. As you can see, if we plot the relative separations between loop extruders in S versus A and B, we get this nice prediction for what the enrichment within S is. Next, if we look at the off-diagonal enrichments between pairs of S, interestingly, we find that this is very well described by the overall abundance of loop extruders in S. Finally, we find that the overall smoothness depends on the overall degree of AB compartment strength, requiring relatively low AB compartmentalization in order to generate such smooth maps. So from each of these features, we understand a different set of parameter space in which the Bombyx-Morai genome might be operating. So if we integrate all of these different parameter estimates, we can generate a model that recapitulates the high C, both visually in terms of this map, where here on the bottom left, we're looking at the best polymer model, and on the top, we're looking at the actual experimental data for this region. And also in terms of the observed over expected enrichments for each type of compartment, as well as the S enrichment within each domain. We also found this interesting and unexpected behavior for the overall 3D organization of S. As I mentioned at the beginning, compartmentalization is reflected as the overall spatial segregation of different types of chromatin. And given loop extrusion's role in driving the decompartmentalization of S in our models, we asked how that might confer a unique spatial organization. And we found that indeed loop extrusion repositions S to the edges of chromosome territories. So in summary, the silk moth genome has 3 compartments, canonical A and B, plus a third type we call secluded S. We believe that S compartments are formed by abundant loop extrusion and little to no underlying affinity. We have estimated various parts of the loop extrusion process from different features in the high C map. And we found a unique role for loop extrusion in positioning loci within the genome. I would like to thank my labs, which is Leonid's lab, specifically Sasha and Ed for inviting me to give a talk today, as well as the rest of the lab, both current and former, as well as my co-advisor's lab of Job Decker.
How do these S regions interact between chromosomes in the genome-wide maps?
Yeah, so despite finding no preferential contacts within cis along a chromosome, we actually find increased interactions in trans, which might correspond with this unique positioning of S at the edges of chromosome territories.
Well, it's an interesting talk. And it's interesting, all these things, all these pictures that you have to have what we call the ideal chromosome plus the phase separation comes very clear from your model. However, this term, this compactation term, this sort of ideal chromosome term that you show here, that's consistent with the data, is probably due to loop extrusion, but it could be something else, right? The data doesn't tell you what the mechanism is.
Yeah, but what we find is that we need something that is capable of generating lengthwise compaction.
That's correct. That's correct. But I think that's a very interesting question in a term that's basic. Loop extrusion appears to be a great candidate to help with this lengthwise compaction, but just the data doesn't give you that. I think that's an interesting thing. It was a very nice talk.
Thank you.
It was indeed a very nice talk. But you told us that there's 3 compartments, and one of the 3 compartments is not caused by compartmentalization. That's a little bit of an inconsistency in language. So do we want to call this compartment S really compartment or not?
It's a good question. How should we describe this? Are each like S a unique microphase?
But it is not a microphase, right? It's not a property of the chromatin. It's a property of having loading sites for extruders and stopping sites for extruders. So in a sense, it's a very different thing from a compartmentalization, right? So we have come to an agreement that compartmentalization is caused by epigenetic markings, and it acts in a certain way that is across space and not along polymer. And now you have a fissure, which you know is not caused by that phenomenon, and you call it with the same name. So I would suggest not using that name.
Yeah, I think I agree with what you're saying. I think we've given it this name given the sort of spatial positioning that we see of these loci, and maybe that's more agnostic to the underlying mechanism of phase separation. But yeah, I think naming things is a difficult thing in our field and should be taken should not be taken lightly. Yeah.
Thank you.
That was a great talk. Is there any epigenetic data on these species at all? Like, do you have a sense of like what marks or biochemically like what could be happening in this secluded compartment as opposed to like A and B, where we kind of know that they biochemically act one way or another? Is there any sense of that?
Yeah, that's really interesting, and somebody else who worked on this paper is giving a talk in a couple of days, Eloise Muller. She's probably going to go into more detail about this, but they're really interesting from both a genetic and epigenetic perspective. They are relatively gene poor, but they house specific types of genes, specifically developmental transcription factors. They're not repressive, like the genes in there are on sometimes, and they show like a mixture of epigenetic states of both euchromatic and heterochromatic marks.
Actually, going from that, these high-C experiments on these particular individuals from the species, or did you do a bunch of different, because you mentioned developmental genes, right? So were these across different developmental time points, the experiments that you had, or anything you noticed between them?
Yeah, so this data is specifically from this larval stage, but we also have data from adult heads in which we also see S compartments. And also I've found like 6 different data sets on NCBI from different tissues and different developmental stages, and they all show S, but sometimes the positions of S are different.
Thank you.
Nice talk. I had a related question, which is are you thinking this purple region is effectively neutral in terms of its affinity, or is it just heavily mixed by loop extrusion? And if you were able to, say, somehow disable loop extruders, that it would kind of separate into lots of small domains, as seen in like cohesin depletion high-C, for example, in humans?
Yeah. Whether or not there's underlying affinity in S is kind of hard to answer, because if you got rid of extrusion, you would see the compartmentalization, regardless of whether or not there is underlying affinity there. What was the other thing I was going to say? I don't remember. But you can have affinity and still generate S or D compartments. It just needs to be sufficiently low to be screened by loop extrusion.
All right. Well, thank you, Emily, and thank you to all the speakers of this session.